Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute. We are actually learning about quality assurance. In my previous lecture, me and your other teachers tried their best to teach you how you can ensure the quality in your pharmaceutical products. We are continuing our learning and we are actually learning our unit 3 lecture of our this entire learning series. Today, I am going to tell you lots of very important things. I am going to tell you the basic introduction, I will tell you the definition of GLP, this means good laboratory practices, what is the scope, what is the purpose of GLP, then general professions, how organization and personnel can play a big role, facilities and equipments. These things we are going to learn on today. So, let me give you some introduction. So, you have to understand that why GLP is important, why it is come to the picture. You see, in earlier time when science was just developing, on those time the things were not so much clear to people, that what to do, what not to do, how to do and how you can manage the issues when it will arise in the preparation of pharmaceutical products, or how to maintain the quality. With passing of time, it has become very much clear that how you can maintain the quality, what you should not to do, what you should not to do in the lab. And with passing of time, you have seen lots of guidelines, lots of that means instruction rules are actually coming. So, in our India, Government of India, Ministry of Health and Family Welfare, they has notified a draft rules to amend the Drug and Cosmetic Act. All of you are aware that Drug and Cosmetic Act, it is very old. Since 1940 to 48, they have actually developed this act and then the rules. So, they amended recently this Drug and Cosmetic Act and they actually now brought the GLP under schedule L1. So, GLPs are the guidelines for quality control and quality assurance in laboratory testing. If you understand GLP very rightly, then in future it will help you a lot to a successful chemist in your pharma industry. GLP is a set of principles intended to support research or marketing permits for product regulated by the government. If I tell you more, see, many of the times students are actually telling me that sir, we are copying, we are following USA or US FDA. Well, I will not comment on the common understanding, but US FDA everywhere when we talk about the quality, US FDA will come to the picture. You know why US FDA is always come to the picture in every kind of discussion? The answer is that they have started the regulation on pharmaceutical products much earlier than any of the country exist in the world. They have formed FDA, I think near about the 1904, and then US FDA regulated the market in terms of its quality, in terms of the safety, in terms of its efficacy. There is a very rigorous rule. US FDA with passing of time, they have tried their best to bring those rules, and we have seen there is something called thalidomide tragedy. I hope all of you might have studied about the thalidomide tragedy. One scientist, one great US FDA chief, she actually saved millions of US people from another tragedy after she has not given approval of the drug. If the drug is in the market, then again they are doing surveillance, post marketing surveillance, we call it as a pharmacovigilance, and due to that they can maintain a check and balance. So, that is why US FDA is always come to the picture and they given much more rigorous definition or rigorous norms, rules for GLP. The formal concept of GLP was first evolved in US in 1970 because of concern about the validity of the preclinical safety data submitted to the FDA, that is Food and Drug Administration, in the context of new drug application. I hope all of you are aware that there are multiple stages are actually associated in drug discovery and drug development. In drug development, first we are actually making some formulation, then we are doing some preclinical studies, CLINICL, then we are doing some clinical trial, there are 3 phases, phase 1, phase 2 and phase 3, and after the defeaters pass, then FDA will approve. FDA will approve. Okay. So, what was happened in 1970 that the pharma companies, they were, when they are actually submitting the safety data to the FDA, that means after preclinical studies, after if you are successful in preclinical studies, that is rat, mice or other species, then you will be filing NDA, new drug application, to FDA. They will give the approval of NDA, and then yes, this is called actually IND, IND and NDA. So, after getting the approval, so the problem was arised in this scenario, they are about the validity of the preclinical safety data. After doing all those preclinical studies, phase 1, phase 2, phase 3, whatever the NDA they are actually submitting. See, this is IND, okay? This is IND and this is NDA. Whatever the NDA they are actually submitting to FDA, there was some issue of validation. At that time, the GLP came to the picture by US FDA. So, what is the definition of GLP? So, good laboratory practice is the quality system concerned with the organizational process and the condition under which preclinical health and environment safety studies planned, also performed, monitored, recorded and also achieved and reported. Okay. So, it is a quality system concerned with the is a quality system. This is such a concept or a system that you can that is bring to your company, bring to your industry. GLP can also be applicable in our labs. If you are doing some research activities in our lab, then GLP also can be applicable. You have to ensure the GLP, because it is a quality system. It is a quality system within the organizational process, and the condition under which the non-clinical or environmental safety studies are actually planned, performed, monitored, recorded and achieved and reported. So, what is the scope of GLP? GLP should apply to all non-clinical safety testing of the test item, for example, like that is natural or biological origin and living system contained in the pharmaceutical products, pesticide products, cosmetic products, veterinary drugs, food and feed additives, also industrial chemical. So, in every cases GLP is actually applicable, because GLP actually helping to minimize the various kinds of your risk, I can say, or it can minimize a lots of issues that we actually face. Okay. So, what is the purpose, why we are actually doing? The purpose is to promote development of quality test data, also comparable quality test of test data to avoid any duplicative test, to avoid the creation of technical barriers to trade, also improve protection of human health and environment, and it can also help scientists to obtain results which are reliable, repeatable and audible. You see, in my previous lecture, I hope you can remember, I have told you about the some stories where in some of the textile companies or some of the pesticide making companies, the health worker, they are actually become sick, they actually become sick. Just a second, they become sick. You know why? Just a second. Just a second, there are some issues. whatever I was telling you that in many of the industries, the people, the working people, they become sick. You know why? Because they are not maintaining GLP and they are not trying to analyze the risk factors. During cleaning, they are using much more lethal substances which may kill microbes, that is fine, but that lethal substances are also equally harmful to our various human cells. Our cells can get mutated, and also if you inhale those things, there are chances your lung cells can be also get damaged. That is why it is a big issue. It was in a research, it was found that those are working in the textile industry or pharma industry, specifically the inhaler based therapeutics or the substances which are actually taken through inhalation, their lung actually get damaged with passing of time and their survival rate actually reduced. So, if you apply GLP, those kinds of issues can be, those kinds of problem can be minimized. Scientists are nowadays they are doing their best to improve the GLP. There are lots of new guidelines, and this can also give you good results. See, if you do perform a work, leave your industry, if you do perform a research work in your lab, ultimately you have to publish this research paper. So, to publish your research paper, you have to give the very data, correct data, and that scenario, reliable data, repeatable data and multiple data can be helpful due to write your research paper. So, this is the purpose of your this thing called GLP. Now, come to the next thing, what are the general provisions of GLP? So, in general provision there are multiple things like scope, then you have definitions, various definition they have mentioned in that GLP provision. Okay. Then during inspection what kinds of testing facilities should be there, it is also mentioned. Okay. So, let me tell you one by one whatever the actual scope they have actually written in the GLP provisions. These parts actually prescribe the good laboratory practices for conducting non-clinical laboratory studies that support to intended to special application for research or marketing permits for product regulated by the FDA, Food and Drug Administration. In definition, they have mentioned a lots of that is a definition, official definition. You can use this definition for your exam purposes, because these are the official definition. Act means Federal Food, Drug and Cosmetic Act. In our India, we have only Drug and Cosmetic Act. Okay, this is for our India, हमारा प्यारा भारत, but this is actually associated with the USA, United States of America. Control article means somewhere if you see the original GLP document prescribed by WHO, in control article means some of the areas you will be observing the word called control article. It means any food additives, color additives, drugs, biological products or any article other than test article for the purpose of establishing the basis for comparison within the test article. Control article versus test article, control article versus test article. Here the word is article, you should not confuse with the article means manuscript, no, not this. Article means substance, it might be some drug substance, it might be some biological substance, it might be or another article or another substance, then the test article. Control means this is the actual, this is the you can compare with this. Okay, this is the ideal you can say in many of the scenario. You can test many of the scenario will not say ideal, but you can compare you test substances with this. For example, let me just remove the ink, highlight these two words. For example, you are trying to test one drug, that drug you have developed for some anti-cancer activity. So, you need some control. So, for example, you have rat, two rats, rat number 1 and rat number 2. you have two rats, rat number 1 and rat number 2. Both the rats having similar exposure or similar amount of cancer. Okay, the cancer whatever it has a similar. Now, you have developed one drug, you may call it as a test, and another. So, what you will do? You will check control that is here and test here, and then you compare. So, for control you have taken one marketed drug, you have taken one some marketed drug. Okay. For example, if I can tell you, you have taken one marketed drug, for example, imatinib, and you have got the IC 50 value, and here again you have taken your test drug, that is new drug, and here you will be getting some IC 50 value. This case you got the IC 50 value, for example, like 50, and here you got the 55. So, this comparison between this, okay, that is actually trying to tell here. Sorry, just a second. Okay, yeah. Now, another thing is the test article. Test article can include anything: food, color additives, drugs, biological substances, etc. Whatever I have already mentioned about these things. Then non-clinical laboratory, another term they have actually given. So, the study means in vivo or in vitro experiment in which a test article are studied prospectively in test system under laboratory condition. Sponsors means a person who initiates, supports by provision of financial resources, and testing facility, it means person, premises or operational units that are necessary for conducting non-clinical and environmental safety studies. Now, another thing is the inspection of the testing facility. I have told you what is testing facility here. Now, how to inspect the testing facility? So, testing facility shall permit an authorized employee of FDA at reasonable time and in a reasonable manner to inspect the facility, to inspect the records and the specimen required to be maintained regarding the studies within the scope of the part. Now, come to the organization and personnel, how GLP actually have defined. So, each individual engaged in the conduct of the responsible supervision of a non-clinical laboratory study shall be education, training and their experience and combination of all to enable that individuals to perform essential function. There shall be sufficient number of personnel for the timely and proper conduct of the study to the protocol. Testing facility, how it will manage? You have seen the personnel. Now, another thing you have to see the testing facility management. We already have seen what is testing facility here. So, for each non-clinical laboratory study, testing facility management shall designate a study director before study is actually initiated, replace the director promptly if he become necessary to do so during the conduct of the study. Assure that there is a quality assurance unit, very important, and assure that test and control article or mixture have been appropriately tested, identity, strength, purity and stability. Now, let me tell you about the study director, because we have mentioned here about the study director for 2 times. So, after learning our about the study director, now let me tell you in details about the study director. So, for each non-clinical study, a scientist or other professional person who has appropriate education, training and expertise will be required. Yes, the study director shall ensure the experiment data, including the observation of unanticipated responses of the test system, are adequately recorded and the systems are specified in the protocol. If I tell you more, the quality assurance unit, the testing facility shall have quality assurance unit which shall responsible for monitoring each study to assure management that the facilities, equipment, personnel, method, practices, records and control are in conformance with the regulation. The quality assurance unit shall maintain the copies of all protocol pertaining all the non clinical laboratory study for which the unit is actually responsible. Facilities, what are the facilities? If I tell you in general, for testing facility, there shall be a suitable size and construction to facilitate about the proper conduct of non-clinical studies. It shall be designed so that there is a degree of separation that will prevent any function or activity from having adverse effect to the study. Animal facility, it is very important. A testing facility shall have sufficient number of the animal house room as in the area needed and assure proper separation of the species of the test system and isolation of the individual projects quarantine. A testing facility shall have number of animal rooms, whatever I have already mentioned, including you have to understand that if there is any biohazards, volatile substances, aerosols and radioactive material is present, then in that case you have to take a special control. Okay, that is why isolation. Then if I tell you more, separate areas will be provided and appropriate for the diagnosis, treatment, control should be also required. And when animal actually are actually housed, facilities shall exist the collection, disposal of animal wastage and reuse of the safe sanitary storage and waste before removal of the testing facility. Supplies, what are the supplies will you provide? There shall be a storage area as need for food, that is feed, bedding, supplies and equipment. Storage areas for feed and the bedding shall be separated from the areas housing for the test system and shall be protected against the infestation, contamination and perishable supplies shall be preserved appropriate means. And facilities for handling test and control articles are necessary to prevent contamination or mix up. There shall be separate areas for receipt and storage test for the test and control articles. If I tell you about the storage areas for the test and control article and the test and control shall be mixture shall be separated. Okay, everywhere you are observing the separation to preserve the identity, strength, preserve and other articles in the mixture. Laboratory operation areas where you will perform your laboratory activity, separate laboratory space will be provided as needed for the performance of routine and specialized procedure required by the non-medical, non-clinical laboratory studies. Specimen and storage data, space shall be provided for achieve limited access of authorized personnel only for storage and retrieval of the raw data and specimen for the corporate studies. And if I tell you the equipment design, equipments are to be used in the generation, measurement and assessment of the data. Equipment used for the facility environment shall be controlled in an appropriate design and adequate capacity to function according to the protocol and shall be suitably located for the operation and inspection. Maintenance, how will do the maintenance? Maintenance shall be adequately inspected, cleaned and maintained. Equipment used for the generation, measurement or assessment of the data shall be adequately tested and calibrated and standardized. The written SOPs, that is standard operating procedure, shall be set forth in the sufficient detail with the method and material and schedule to be used in the routine inspection, cleaning, maintenance, testing, calibration, standardization of the equipment and also shall specify when appropriate and remedial action should be taken. Okay. There should be one proper SOP where everything will be written. Maintenance and calibration, written record should be maintained for the inspection. See, it is very much important that you should maintain the written record, because by observing all the record you will be able to understand that where you have done the previous mistakes. Okay. So, in our today's lecture we have learned about the introduction. I have told you about GLP, what is the scope of GLP, you also have seen the purpose of GLP, what are the general provisions, you have seen the organization and personnel under GLP and what are the facilities, equipments, how it should be and how it is playing a big role. These things today we actually have covered. In our next lecture tomorrow, I am going to tell you about the testing facilities operation. I am going to also tell you about the testing control articles. Okay. So, that is it from my side. Thank you all of you for joining today's lecture. I wish all of you are learning in a very great manner. Your understanding for this subject is increasing a lot. So, thank you all of you. All the best. We shall meet in our next lecture.